Hello students, welcome to Vedantu Karnataka. Hegidira ellaru chennagidira. So today we are going to start our first class for all our 12th CBSE students. So we got a lot of requests, ma'am, why are you doing only PUC? Please do CBSE also. So we thought why not start CBSE for all of you. So today is going to be our first class for CBSE and I want all of you to share with all your neighbors, relatives who are right now in class 12 CBSE and who is looking for a proper channel that explains everything for them. Okay, so today we are going to start sexual reproduction in flowering plants. We might be doing it in parts today because this chapter has 3 parts, pre-fertilization, fertilization and post-fertilization. So we will divide and do the chapters, okay, like the topics. So students, before that I want to inform all of you on 21st, on Saturday, sharp at 6 p.m. there is going to be a VAL function where we are going to felicitate our achievers who has done who has brought so much happiness to Vedantu with their success in NEET, JEE and Olympiad. So I want all of you to stay tuned to the channel sharp at 6 p.m. And click on the notification and click on the bell icon so you do not miss that, because it's going to be motivating for you to see your seniors achieving so much and you will be like, yes, next year I want to be there. ಸೊ ಅದಕ್ಕೋಸ್ಕರ ಎಲ್ಲರೂ ಕರೆಕ್ಟಾಗಿ ಶಾರ್ಪ್ 6 PM ಸ್ಯಾಟರ್ಡೆ ಖಂಡಿತ ಚಾನಲಲ್ಲಿ ಬರಬೇಕು. ಈ ಲೈವ್ ನೋಡಿ ನಿಮ್ಮ ಸೀನಿಯರ್ಸಿಗೆ ಆ ಒಂದು ಅಪ್ರಿಸಿಯೇಷನ್ ಕೊಡಬೇಕು. ಓಕೆ, ಸೊ ಎಸ್, ನಾವು ವಿ ವಿಲ್ ಡೈರೆಕ್ಟ್ಲಿ ಸ್ಟಾರ್ಟ್ ದ ಕ್ಲಾಸ್. ಸೊ ಸ್ಟೂಡೆಂಟ್ಸ್, ಜನರಲಿ ನೀಟಿಗೆ ಬಂದ್ಬಿಟ್ಟು ಈ ಚಾಪ್ಟರ್ ಒಂದು 5 ಮಾರ್ಕ್ಸ್ಗೆ ಬರುತ್ತೆ, ಅಂದರೆ 5 ಕ್ವೆಶನ್ಸ್ ಬರುತ್ತೆ 20 ಮಾರ್ಕ್ಸಿಗೆ. KCETಲೂ 5 ಮಾರ್ಕ್ಸ್, 5 questions come from this. So CBSE exam also, every chapter is important. There is nothing called as, high weightage, low weightage in the examination. Now, talking about sexual reproduction in flowering plants is one of the most important concepts. Now here you have to learn few things. Okay, what are the most important things that you have to study? You have to study about what is sexual reproduction. Why is sexual reproduction necessary, right? So sexual reproduction. What is the meaning of sexual reproduction, students? Whenever the word sexual comes, that means there is going to be variation. Okay, because why? Because there is involvement of gametes. Okay, and gametes are going to be male and female gametes, right? Male and female gametes that will allow us for variation to happen. So sexual reproduction word itself talks about variation, and reproduction, where fertilization is going to happen, male and female gametes are going to come together to form the zygote. Now, why is it necessary for the plant to do sexual reproduction? Alva, that is the question. So, illi plants sexual reproduction tumba important so that they can also ultimately form seeds, fruits. The ultimate destination is going to be formation of seed, formation of fruit. How are they going to form this? With the help of here ovary fertilized. Here we have ovary, here we have fertilized ovule. So these people are going to help, okay? Arrow mark. These people are going to help us for that, okay? So in this chapter, you are going to basically study about that. See, sexual reproduction chapter, like when you are trying to study it, understand, sexual reproduction in plants does not mean that Chintu can give flower to Mintu. It's not about that one day. Flowers are doing it so they can do pollination, they can form fruits, they can form seeds. That's the ultimate goal. Clear to all of you? That is the ultimate goal of doing this. So that is your introduction here. Now look here, students, NCERT lines nodana solpa. So are we not lucky that plants reproduce sexually? The myriads of flowers that we enjoy gazing at, the scents, the perfumes that we spoon over, the rich colors that attract us are all an aid of sexual reproduction, right? Fragrance, we enjoy the fragrance of flower, that is also because of sexual reproduction to attract the insects. We look at the inflorescence, that is one flower will be very small. So a group of flowers, when we look at them and when we look at them, we want to gaze at them, admire them, take photos of them. All that is because of sexual reproduction, okay? Now, all flowering plants, it's tumba important. All flowering plants show sexual reproduction. All flowering plants show sexual reproduction because in a flower, what do you see? You see the male and the female part, right? Now, here other things are given, don't have to worry, but look here. range of adaptation to ensure formation of the end products, okay, the end products of sexual reproduction. What is the end product of sexual reproduction, students? The fruits and the seeds, okay, the fruits and the seeds are going to be the end product of this. Clear? Yes, easy peasy lemon squeezy. Now, to a biologist, okay, to a biologist, flowers are morphological and embryological marvels and sites of sexual reproduction, right? That is the site at where the sexual reproduction is happening. Correct or not? Yes, ma'am, it is correct. Now, in class 11th, you have read the various parts of flowers, morphology of flowering plants, ಸ್ವಲ್ಪ basics ಎಲ್ಲ ಓದಿರ್ತೀರಾ. So, which will help you recall the parts of a typical flower, but still we will draw and learn the flower. Okay, before that, students, see, to a biologist, flower is a morphological and embryological change they have mentioned. Now, you have to understand what those words are talking about. See, look here. See, normally, students, this is your shoot. This is the shoot, correct? On the shoot, there are some shoot apical meristems. What is that? SAM is there, shoot apical meristem. The shoot apical meristem will differentiate and it will slowly start forming, okay, slowly start forming it will help in the formation of your floral meristem. It will help in the formation of the floral meristem. So, shoot apical meristem is now becoming slowly the floral meristem. That means when it was SAM, okay, SAM's main job was to form the leaves, right? Now, here floral meristem's job will be what? To do the changes to form ultimately the flower, correct? To ultimately form the flower. Clear to all of you? To ultimately form the flower. In between this, you can also write, students, floral primordium. It's like an organ, okay? Primordium. So, from here, we have finally got the floral meristems. Okay, clear to all of you? Now, talking about, this is how the flower formation happens. So, before the actual flower formation, please do understand that plant has done two things. Plant has already changed themselves structurally. Structural changes can be seen. Can you see structural changes? Yes, ma'am, structural changes. And not only that, how did this happen? Because of hormonal changes. Hormones are released, okay, hormonal changes. So, structural changes and hormonal changes have already taken place. These two things have already taken place before the actual formation of the flower. So, students, very easy. ನೀವು ಇವಾಗ CBSE 12th board exam ಬರೆಯಕ್ಕೆ ಮುಂಚೆನೆ ನೀವು already prepared ಆಗಿರ್ತೀರಾ that I want to score this much, 95%, 96%, I want to do this, after this I want to write KCET. So that's a plan. That means actually before writing the exam itself, you have already made a plan what you want to do. That is what we are talking about here. Clear to all of you? Yes or no? Okay, one second. Yes, that is clear. I hope all of you are making notes. Students, ee notes ella nimige Vedantu Karnataka Telegram channel allo, Gopika Biology official allo, you will be able to get all of this, okay? Yes. Done. Okay, perfect. Okay, got it. That's why to a biologist there is a structural and hormonal changes. Now, structure of a flower. Let's first draw a beautiful flower and then understand the parts of the flower, okay? So, I am starting off with, students, the stigma, style and ovary. So, whichever chapter you want next, please let me know in the comments and also share it with all your CBSE classmates who are suffering, after college to understand topics. This channel will help you with PCMB entire content, okay? So, here students, nodi, stigma, style, ovary I have drawn. Now, here I am going to draw sepal and then a small swollen portion. Same thing continues here, like this and like this, okay? Yes, this is the portion that you hold and this is the portion where the swollen parts are seen, okay? Now, to this I have to draw some beautiful long anthers, okay? like this, anthers, okay, because they are bilobed, okay. You can draw more than 1 or 2 students, okay. Yes, and now we will complete it with the help of some flowers, petals. Okay, some petals, okay, flower looks kind of incomplete, so we will just draw this, okay, good. This is your flower. Now let's label each of the part and understand which is essential for us, which is non-essential for us. Now look at this students, this is going to start, first portion that you are seeing here is an anther. What is it? An anther, okay, and this long stalk like structure that you are seeing is filament. What is it? Filament, okay. Then when you come here, obviously this is petal. Then you come more here, you will be able to see stigma. I don't think I can remove this, okay. This is your style, okay. This is the stigma. This is the portion where the pollen grains are going to land, okay. Stigma, style, and this is going to be the ovary. Inside the ovary, who will be there? Ovules will be there. Okay, inside the ovary, ovules will be there. So this side is labeled, done. Okay. Now here students, this green color portion that you are seeing is called the sepal. Okay. This swollen portion is called thalamus or thalamus, also called as receptacle. Okay. And this portion that everyone holds and gives no, take please accept. Okay. So that is your pedicel. Okay, pedicel. So this is the portion. Did we miss anything? Anther, filament, stigma, style, ovary, ovule, sepal, thalamus, pedicel. Awesome. Okay, we have done everything. Now students, I will give you some important things here. Anther and filament together is called stamen. Anther and filament together is called stamen. Okay. Now let's say if we label stigma, style, ovary together. Okay, I don't know, I'm drawing a building, I feel now. Okay, this is going to be called your carpel or pistil. Okay, pistil, right? Stigma, style, ovary, even your ovules are going to be included in that. Now students, pedicel, thalamus, receptacle, sepal. Now we will come into this. Students, please understand that flowers have 4 whorls. What do they have? 4 whorls. Okay, whorls, not world. Okay, 4 whorls. First whorl is going to be called the calyx, which has many units of sepals. So first whorl that you are seeing is going to be having many units of sepals. That is called as what? Calyx. Okay. Adamele illi you will see corolla. Ill students, many units of ಏನು ನೋಡ್ತೀರಾ? Petals ನೋಡ್ತೀರಾ. Okay. Then you have andrecium. Andrecium is going to be what? The male part, right? This is going to have many units of stamens. Many units of stamens. Okay. And then here you have gynoecium. That will have many units of carpel or pistil. Clear? So these are the 4 whorls which are very, very important for you. If you know 4 whorls: calyx, sepal, corolla, petal, andrecium, stamen, gynoecium, carpel. So now students, illೊಂದು differentiation ಮಾಡಬೇಕು. ಏನು differentiation ಅಂದ್ರೆ flower, for a flower what is important? Okay, for a flower what is really important is the question. Okay, for a flower what is more important? That is what we are going to understand. So for a flower it needs some essential parts. Essential. Now what do I mean by essential? That means the ones that are actually going to help with the fertilization. Okay, so essential is going to be none other than your androcium or andrecium and gynoecium. Andrecium and gynoecium. Now andrecium students, it will have what? It will have the stamen, right? It will have the stamen. Stamen, when I am saying, always remember I am talking about anther and filament, okay. Here gynoecium will have carpel. Here I am talking about stigma style ovary. So this becomes the essential part. Then ma'am, does something called as non-essential part exist? Yes, it does exist. Non-essential part, students, it's going to be the one who is not directly involved in fertilization, but they are part of it. They are going to help with it. Non-essential part will be what? Your sepal and your corolla, or calyx and corolla. Clear? Non-essential means what? This sepal actually protects the bud stage. Okay, when the flower is in a bud form, you will see that the entire thing is covered in green color. That is because the sepal is protecting. Now petal, when I talk about, insects are getting attracted because of the petals, because of the beautiful colors of the petal. So our directly fertilization ge help ಮಾಡ್ತಾ ಇಲ್ಲ, ಅಂದ್ರೆ gamete formation ಗೆ help ಮಾಡ್ತಾ ಇಲ್ಲ, but indirectly pollination time ಅಲ್ಲಿ help ಮಾಡ್ತಾರೆ. Okay na? So this is your entire flower. I hope all of you are understanding. Please give me a thumbs up if all of you are understanding. Okay, that will help me have a picture. Okay, now students, what are the events in reproduction? Okay, events in reproduction is going to be three major events. First event is going to be your pre-fertilization. Pre-fertilization. Idu madve tara, marriage tara. Pre-shoot, pre-wedding shoot, post-shoot, during the wedding shoot, ಆ ಥರನೇ pre-fertilization events. ಅಂದರೆ how do we form the gamete? Fertilization, how does the gamete meet each other? Post-fertilization, how does the ovules and ovary, how does they form seed and fruit? Okay, so pre-fertilization, then we have fertilization, then we have post-fertilization. Post-fertilization. Clear? Pre, post and fertilization. So pre-fertilization, students, what is exactly going to happen? In pre-fertilization, two events are going to happen. Microsporogenesis, megasporogenesis. Microsporogenesis and megasporogenesis. Microsporogenesis andre students, ಇಲ್ಲಿ ನಾವು male gamete formation ಮಾಡ್ತಾ ಇದ್ದೀವಿ. Okay, male gamete formation. ಇಲ್ಲಿ ನೋಡಿ mega, who does big dramas, mega dramas. Okay, so here we are going to do female gamete. Female gamete, okay. After this lecture, you just have to read your NCERT once. You will be thorough with every single topic, okay. In fertilization time only, students, here we are going to discuss about double fertilization. Double fertilization. In double fertilization, ಎರಡು topics ಓದ್ತೀರಾ, syngamy ಮತ್ತೆ triple fusion. Triple fusion. Done. Syngamy plus triple fusion. Post fertilization ಅಲ್ಲಿ ಏನು ಓದ್ತೀರಾ? Seed formation ಮತ್ತೆ fruit formation. Seed formation ಮತ್ತೆ fruit formation. That's what you're going to study here. Clear? So this is how the chapter division is going to happen. All of you can take a screenshot, or all of you want to write it down, please take a minute and write it down. No issue with that. Okay, no issue at all with that. Done? Okay, perfect. I think all of you wrote it down. So this is the events in reproduction, okay. Now ill nodi students, pre-fertilization, here they have given NCERT lines. So much before the actual flower is seen on the plant, the decision that the plant is going to flower has taken place. Several hormonal, see, I told you, several hormonal and structural changes are initiated, which leads to differentiation and further development of the floral primordium. Okay. Now, inflorescence are formed which bear floral buds. Inflorescence andre just imagine ಮಾಡಿ ಒಂದು ಫ್ಲವರ್ ಇಷ್ಟೇ ಇರೋದು ಸ್ಟೂಡೆಂಟ್ಸ್, ಇಷ್ಟೇ ಇರೋದು ಫ್ಲವರು. ಈ ಫ್ಲವರ್ನ ಇನ್ಸೆಕ್ಟ್ ಅಟ್ರಾಕ್ಟ್ ಆಗುತ್ತಾ ಇಲ್ವಾ? ಇಲ್ಲ ಮ್ಯಾಮ್, ಅಟ್ರಾಕ್ಟ್ ಆಗಲ್ಲ. ಸೊ ಅದಕ್ಕೆ ನಾವೇನು ಮಾಡ್ತೀವಿ? ಸೊ ವಿ ಡೂ ಲೈಕ್ ದಿಸ್. ಓಕೆ, like this. Now will the insect get attract? Obviously ma'am. Will insect attract this or this? It will obviously attract this. So this is an inflorescence where a group of flowers come together. Okay, so inflorescence are formed which bear the floral buds and then the flowers. In the flower, the male and the female reproductive structures, the androecium and the gynoecium, differentiate and develop. You would recollect that androecium consists of a whorl of, see, androecium consists of a whorl of stamen representing the male reproductive organs, and gynoecium represents, okay, represents the female reproductive organ. Clear? Done? Female reproductive organ. Okay, perfect. Now moving forward, androecium. Okay, we're going to start the main portions. So that's why I want all of you to listen carefully. Okay, listen carefully. Okay, come on. Androecium. Okay, androecium. So look here students, when I talk about androecium, first thing what are you thinking about? Ma'am, we are talking or thinking about anther and filament, correct? Anther and filament. Now, this anther and filament students, sometimes they can be attached to the petal or they will be attached to the thalamus. Okay, I am just drawing like this now, no time to draw the whole thing. Okay, like this. Now, they say in your NCERT, the portion that is closer, the portion that is very closer to the thalamus is going to be the proximal end, and the portion that is very far from the thalamus is going to be called the distal end. Okay, that means what? The one that is very close, the one that is very close to the thalamus is going to be called your proximal end, and the one that is very far is going to be called the distal end. So on the distal end from the thalamus we can see the anther, and in the proximal end we can see the filament alva. Ivaga ನಾನು ಇಲ್ಲಿದ್ದೀನಿ, ಕ್ಯಾಮೆರಾ ಅಲ್ಲಿದೆ, ಸೊ ಅದು ನನ್ನ ಡಿಸ್ಟಲ್ ಎಂಟು. ಈ ಬಾಟಲ್ ನನಗೆ ತುಂಬ ಕ್ಲೋಸ್ ಆಗಿದೆ, ಸೊ ದಿಸ್ ಇಸ್ ಮೈ proximal end. So this is the proximal end, camera is the distal end. Clear to all of you? So that is how you have to remember. Clear? Now this one line they have mentioned in your NCERT. Now students, if I further take the anther, okay, I just take only the anther, like a big portion of the anther, just magnify it and show it to all of you. This is how your anther is going to look, bilobed anther. Okay, so instead of this we can just draw like this also. Okay, like this it will look, kind of looks like a peanut, but then yeah, look at this, this is the bilobed anther, one lobe, okay, and another lobe, one other lobe. Done? Okay, done. So this is how. Now see, from here the filament is going to arise. Okay, filament is going to arise. Now look here students, if I take a cross section of this, if I take a transverse section like this, I am going to get something like this. I am going to get something like this. All of you notice clearly. See, I will have a portion like this because this is cut. Okay, something like this. I will have one more portion which is cut, and that will also have something like this. Okay, now I cut it down, no? So here there is going to be a space and something like this. Here also there is going to be a space, something like this. Correct? If I cut from the center, something like this is what I am going to get. Now students, if I make this like this, okay, just imagine this two I am holding in this hand, this two I am holding down. If I make it like this, how is it going to look? It is going to look like this. Like this, correct? Yes or no? Yes ma'am, it's going to look like this. Okay, I want all of you to understand the diagram, then only the entire concept will be clear. So if I turn it this way, you are going to get something like that. Okay, it looks like an amoeba, butterfly and all, but it's not. So here you understand that these people are two lobes. Okay, lobe 1, okay, lobe 1 and lobe 2. So we say that our anther is bilobed. What do we say? We say our anther is bilobed. Okay. Now, we will go further into understanding the anther diagram, but I want all of you to understand this. Now, students, in the center, okay, in the center if you look, there is one line like this, right, there is one line like this, which is actually a little more thicker and broader. This line is called line of dehiscence. What is it called? It is called the line of dehiscence. Okay, I will draw it with another color because it is not visible. So, here you have a line which is called the line of dehiscence. What is line of dehiscence meaning? After the pollen grains are formed inside it, it has to break open to release the pollen grains. Otherwise, how will the pollen grains go outside? So, that makes the line of dehiscence. That makes the line of dehiscence. Clear to all of you? Line of dehiscence. Clear? Yes. Okay. Awesome. Now, continuing. Structure of anther. So, students, that structure which I turned, no, like this, the structure which I turned, we have to draw the magnified image of it. So, let us go. The same diagram given in your NCERT. Okay. So, it does not have to be properly shaped, okay, it does not have to be, it is kind of, you know, irregularly patterned. Yeah. So, if you look at this, okay, look at this carefully, you notice few things, right. You notice that, ma'am, these are the lobes, right, this is one lobe, this is one lobe. So, that is why these people are called bilobe. Now, students, inside this lobes, there are small portions, okay, inside this lobe, there are small portions which are called as the theca. They are also like a lobe only, like one space that is given for the microsporangia to sit. So, this is one theca where I am placing the microsporangia, okay. This is another theca where I am placing the microsporangia. This is one more theca where I am placing the microsporangia, and this is one more theca where I am placing the microsporangia. So, that means in one lobe, okay, in one lobe you will have two thecas, correct? In one lobe, two thecas. So, we call this condition as dithecous. What do you call that? Dithecous. So, in one lobe we have two thecas. Done? Is this clear? I hope this is clear. Now, to keep both the lobes together, to keep both the lobes together, students, there is in between, there is a structure which is called the connective. Okay, this one, this structure is called the connective. I am just drawing the shape, okay, it is actually circle shaped also, you can draw. So, this is a connective which keeps both the lobes together. Now, important things, what is this pink color structure that I have drawn? It is called as the microsporangia. What it is called? Microsporangia. So, what are the two processes that happen here? Microsporogenesis. So, microsporangia is going to bear your sporogenous tissue or your microspores, so that can ultimately form the pollen grains. We will learn about it, don't worry. Now, students, if I take a magnified image of this microsporangia, it will have 4 layers to do a protection, okay. So, the first layer, understand the first layer is common, okay, common means it is running throughout the anther. That is going to be a first layer like this. This layer is called the epidermis. What is this layer called? Epidermis. I will just fill these gaps. Yes, epidermis. The first layer that I have highlighted is called the epidermis, okay. I will just draw it with that, I will label it with that color only, so it becomes easier. Epidermis, okay. Now, inside after that we have one layer which is called your endothelium, again common for all of them, okay, we have common for all of them. So, again I am drawing just one layer like this. These are all single layers, okay. We have a endothelium, okay. Can you see the green? Yes, it is visible. I will make it little thick if you want, because I want all of you to remember the colors. Students, always using colors helps in our studies because visual presentation is the best way to remember, okay. So, this is going to be your endothelium. endothelium. So first two layers are done, epidermis and endothelium. Okay. Now inside we will have middle layer, which is individual for all the microspores. Okay, it is individual for all the microspores. Middle layer, students, remember it can be more than one layer. Okay, it will not be one layer, it will be more than one layer. So I will just maybe like draw cells types. Like this, okay. Middle layer will be more than one layer. So I am just drawing two layers like this. Okay, like this. Two layers of middle layer I have drawn. Now this one is the middle layer, which will be individual for each microsporangia. One middle layer, middle layer, middle layer, middle layer. Okay, understood? Now inside this, students, there is going to be one more layer, which is called the tapetum. Which is called what? The tapetum, which is going to do the job of nourishment. Okay, it is going to have the food reserve. It is going to be multinucleated. It is going to provide the nourishment. Okay, tapetum. It is going to provide the nourishment for the growing, pollen grains. Now, remember, in between I told you there are some special cells. Okay, there are some special cells, and these cells are called as your sporogenous tissues. These are the potential to become the microspore mother cell. They have the potential to become the microspore mother cell. See, like this. Clear? Now, here, where do I draw it, students? They are the sporogenous tissue. Sporogenous tissue. Done? Okay? Yes? So I think I will just draw the connective, because too much snake pattern also you people will get confused. So I'll just draw it like this. Okay? Done? Clear? Just look at the diagram for a minute, you will be able to understand. Okay, there's nothing to panic and all of that. It's very simple. Yeah, just look at the diagram. Okay, so first layer is going to be epidermis, which is going to line the entire anther. Okay, second layer is going to be endothelium, again which lines the entire anther. Third layer is going to be middle layer, which is a multi-layer, which is present for each microsporangia. Okay, then after that we have a layer called tapetum, which does the nourishment for the growing pollen grains and growing pollen, and in between you have a special type of tissue which are called as sporogenous tissue, which has a complete potential to become the microspore mother cell, which has the complete potential to become the microspore mother cell. Clear to all of you? Now, let's write about each of the layers. Okay, this is single layer. Okay, it's going to help with protection, so we will come to that. Again, single layer. Okay, this single layer. Now this one is multi-layered. Multi-layered. Okay. Now students, remember 1, 2 and 3. All these people are going to do protection and dehiscence. Okay. Their main job is going to help in protection and dehiscence. Protection and dehiscence. Done? Clear? Now, we will talk about tapetum. Tapetum is going to do the nourishment. It's going to do the nourishment. It is going to do the nourishment for the growing pollen grains, and they have a lot of food reserve, dense cytoplasm. Okay, dense cytoplasm, and they are multinucleated. Okay, some additional information, they have more than one nucleus also I can write, because they can be binucleated as well. They have more than one nucleus. Okay, I hope this is clear. Yes, done. Okay, connective will keep two lobes together. Okay, so here students, diathecus, because there are 4 microsporangia, we call this condition, okay, we call this anther dithecus, sorry, tetrasporangiate, tetrasporangiate, okay. So this is the two words that you use. Other than that, what do you use? Anther is bilobed. Depending on the shape, we can also say the anther is tetragonal in shape. Okay, we can also say anther is tetragonal in shape. Clear to all of you? Okay, done. Okay, perfect. Yes, I have used a lot of colors so that you people will be able to remember everything properly. Okay, I hope this is clear to all of you. Now students, now structure of anther is clear. Now I will talk about the sporogenous tissue. Okay, now I told you that the sporogenous tissue which is there, okay, let's first draw the circle. Okay, this is your microsporangia. Okay, they are going to be right in the center of the microsporangia. Inside that they are going to have few layers of your middle layer. Okay, like this middle layer is going to be there. Okay, like this middle layer will be there, two layers of middle layer, and they will have one layer of tapetum, correct, one layer of tapetum. So I have just drawn blue. Okay, one layer of tapetum is there, correct. Now in between this, what did I tell you? There were sporogenous tissues, sporogenous tissues like this. Like this, clear? Sporogenous tissues there. Done. Okay. Now this sporogenous tissue has the potential. Okay, this sporogenous tissue has the potential to form the mother cell. Okay, pollen mother cell. Why students pollen mother cell? Otherwise, who is going to do the entire process properly? So this sporogenous tissue, these people have the potential to form the MMC. MMC I won't write because it will be confusing with megaspore. Microspore mother cell, okay, and PMC, pollen mother cell. This PMC will undergo meiosis, then only the process is going to continue. So here remember, the PMC will undergo meiosis, okay, and after that what we are going to get? We are going to get microspore tetrad. What are we going to get? Microspore tetrad. Clear? So this is how your microsporogenesis happen, where the microspore mother cell or your PMC, that is your pollen mother cell, will undergo meiosis, reductional division and form microspore tetrad. Okay, now students remember, one second, I'll just mute this. Okay. This microspore tetrad, in the starting, what happens students? These people were all clustered together. These are lines of NCERT. They were all clustered together like this. Like this, clustered together. Okay, this is also because of a callus, okay, which you don't have to know for CBSE also. But this clustering we had to break. For that, tapetum came into help with the help of callase enzyme. Okay, this we teach for the NEET students. Now, this people, after using that enzyme, it got separated. So this separation, okay, this separation led to the formation of separate microspore tetrads. That means each of them became individuals, okay, with gaps, proper gaps, and they can mature to form the pollen grains. Okay, so now this each microspore tetrad students, each of them will start becoming a pollen grain. Each of them will start becoming what? A pollen grain. So let's draw a pollen. Let's draw one pollen grain. Okay, this is the outer layer, inner layer. Okay, it might look like a sun, but trust the process, sun. Okay, so like this, intine, exine, okay, they'll have two cells, one vegetative cell and one generative cell. Okay, like this. Done, all of you. So this is your pollen. So each of them will become a pollen grain. So here what happens, students? Microspore tetrads are there. Their ploidy is going to be haploid N, 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 N. Okay, and then this got separated. After separating, each of them are going to form a pollen grain. Now, students, when microspores were present inside microsporangia, we call it microsporangium or microsporangia. Now, when pollen grains are going to be inside this, we call it pollen sac. Okay, we call it the pollen sac. Clear? This pollen sac has to dehiscend, then only the pollen grains will be transferred. So, this was microsporangium. Okay, when it was not matured. Okay, when maturity happened, we call it the pollen sac. Okay, I hope this is clear. This is what is called as microsporogenesis. This is what is called as microsporogenesis. That means formation of, okay, write the definition. Definitions are important for you people. Formation of microspore tetrad. Formation of microspore tetrad with pollen mother cell. Okay, I think with is the wrong word. Formation of microspores with meiosis is called as, okay, I think with is the wrong spelling. Okay, there should be a word for it. Okay, 1 second. When the anther is young, anther develops, the cells of the sporogenous tissue undergo division to form microspore tetrads. The process of formation of microspores from a pollen mother cell through meiosis. Okay, sometimes English does not English well. Okay, formation of microspore tetrads from PMC, I am writing pollen mother cell through meiosis. Is called as microsporogenesis. Clear? Very important. This is your microsporogenesis for all of you. Okay? Yes. Done? All of you? Pakka? They can form potential microspore mother cell or PMC. Okay, PMC. They will undergo meiosis. Okay, they will undergo meiosis, and that will form microspore tetrads. Done, done, all of you? If you are enjoying so far, all of you quickly smash the like button, okay, and share it with your friends. Let them also study. So, students, pollen grains we will have to study in detail. Don't worry, we will study about that. Okay, right now I hope this is clear to all of you. Okay, this is clear. We will go through NCERT once. So, a typical angiosperm anther is bilobed, okay, and each lobe has 2 thecas. I told you each lobe will have 2 thecas, that is, they are dithecous. Now, often a longitudinal groove runs lengthwise separating the thecas. Let us understand the various types of tissues and their organizations. In transverse section, okay, in transverse section of the anther, the bilobed nature of the anther is very distinct in a transverse section of the anther. Anther is 4-sided, tetragonal in shape, tetrasporangiate also, okay. So, they consist of 4 microsporangia located at the corner of each lobe, okay, 2 in each lobe. The microsporangia develop further to become the pollen sac. Okay, to become the pollen sac. They extend longitudinally all through the length of an anther and are packed with pollen grains. That's why certain times when you take a flower, you will notice that there is some yellow color dusty thing in your hand. That is your pollen grains. Okay, that is going to be your pollen grains. Now look here students, this is the structure. You can see the entire structure. In middle you have the connective which will connect both the lobes. Then you have the outermost layer which is the epidermis, endothecium. Can you see epidermis and endothecium is running all over the anther. Okay, then we have middle layer. Can you see middle layer? This portion like this middle layer is there. Then we have tapetum, which is this layer, okay, tapetum, and then we have sporogenous tissue which is right in the center. Now if you take a magnified image of that, this is how it will look. Epidermis, endothecium, middle layer, you can see multi-layer there. Microspore mother cell, that is again your sporogenous tissue, have the potential to become the MMC, so they will become like that. And then you have tapetum for nourishment, okay, tapetum for nourishment. I hope it is clear. Now microsporogenesis. Okay. Question is, why are the tapetal cells binucleate? Okay. Question is, why are the tapetal cells binucleate? So students, what exactly happens here is the tapetal cell. Let's say this is your tapetal cell. It will undergo karyokinesis. Karyokinesis means what? Where the nucleus divide. Okay, division of nucleus. Karyokinesis will happen, then obviously what will happen to the cell? This will be the cell, it will have now two nucleus. Okay, it became binucleated. But if karyokinesis is not followed, okay, by cytokinesis, then there is no cell wall formation that will happen. So in tapetal cells, what happened? Karyokinesis happened that led to the formation of the nucleus, but cytokinesis did not happen, that led to the formation of binucleate tapetum. Students, this reason could be like why cytokinesis did not happen is to actually for better efficiency. Okay, for better efficiency they do like this. So that is what happened here also, that tapetal cells underwent karyokinesis where the nucleus got divided. Okay, so I am writing it here. Division of nucleus, okay, and cytokinesis division of the cytoplasm. Here majorly what happens? Cell wall formation happened. So karyokinesis not followed by a cytokinesis led to the binucleate stage of tapetal cells. Clear to all of you? Yes. Now pollen grain formation, as you already saw, the microspores, okay, these were our microspores, okay, these microspores developed to form what, students? It ended up forming the, like this, an intine, an inner layer of the pollen grain, and then it formed an outer layer which is the exine, okay. Okay, exine formation happened. Inside that what happened? There was a big cell and then there was a small cell. The big cell had a round nucleus, the small cell had a spindle shaped nucleus, and obviously cytoplasm. Okay, dense cytoplasm was noticed in the big cell, and in that cytoplasm only the small cell was floating. So this is your pollen grain. Now students, let's label the pollen grain portion. Outside what you are seeing is called the exine. This is the outermost layer, and this exine is made up of one of the most resistant material on earth, which is called as sporopollenin. One of the most resistant material called as sporopollenin. Then students, the yellow color that you are seeing is called as intine, okay, which is made up of cellulose and pectin. Cellulose and pectin, okay, intine. Now if you notice here, there is a portion where the sporopollen is not present, which is called the germ pore. Why this is necessary? So pollen tube germination can happen, because sporopollen is one of the most resistant substance. If such a resistant substance is there, is no way I can damage the substance with acid, alkali or anything, right? ಅದಕ್ಕೋಸ್ಕರ ಒಂದು ಸ್ಮಾಲ್ ಸ್ಪೇಸ್ ಕೊಟ್ಟಿದ್ದಾರೆ ವೇರ್ ದ ಸ್ಪೋರೋಪಾಲಿನ್ ಇಸ್ ಆಬ್ಸೆಂಟ್. ಅದಕ್ಕೆ ನಾವು ಅದನ್ನು ಏನು ಕರಿತೀವಿ? ಜರ್ಮ್ ಪೋರ್ ಅಂತ ಕರಿತೀವಿ. ಓಕೆನಾ? Now students, look here, there are two cells. First one is going to be called your vegetative cell. The bigger cell is the vegetative cell. Okay? big cell is going to be called the vegetative cell. The small cell is going to be called the generative cell. Now students, this generative cell will have your male gamete. Generative cell will have your male gamete. Vegetative cell is going to have the food reserve for the growing male gamete, to provide the male gamete. Okay, so it is going to have the food reserve, that's why it has dense cytoplasm. Generative cell is the one that will have the MG, that is the male gamete. Clear to all of you? So this is what a pollen grain will look like, and the sporopollen students, they have beautiful, structure, because of which pollen grains are sometimes ornamental. Okay, they look ornamental, and they can survive however long they want because the material is very, very resistant. Okay, very, very resistant. Done? I hope this is clear. Perfect. Okay, so this is your microspore. Don't worry, these notes will be shared. So this will be one of your best notes that you can have, because I am drawing diagrams for everything and teaching you. Microspore tetrad, this microspore tetrad will form the pollen grain, and once the pollen grain starts being inside the pollen microsporangia, we call it the pollen sac. Okay, pollen grains. Clear? Done? Pollen grain. So this is what has happened. I hope it's clear. Okay, so this is done. Okay, now we can just go through this also very quickly, your NCERT. So in a transverse section of an anther, a typical microsporangium appears near circular in outline. It is generally surrounded by 4 wall layers. You know what are the 4 wall layers? Epidermis, endothelium, middle layer and tapetum. Okay, the epidermis, endothelium, middle layer and the tapetum. The outer 3 layers perform the function of protection and dehiscence. Okay, help in dehiscence of the anther, so they can release the pollen. Okay, the innermost layer is the tapetum, which helps in nourishment. Tapetum nourishes, okay, nourishes the developing pollen grains. Cells of the tapetum possesses dense cytoplasm. Why, students? Because they need to have the food reserve. Okay, they will have dense cytoplasm and generally have more than one nucleus. Okay, they'll have more than one nucleus. Can you think of how tapetal cells could become binucleated? I just explained you. Karyokinesis not followed by cytokinesis. Okay, when the anther is young, a group of compactly arranged homogeneous cells called as sporogenous tissue occupy the center. Okay, occupy the center. They are homogeneous, same. Okay, now center of the microsporangium. Microsporogenesis, as the anther develops, the cells of the sporogenous tissue undergo meiotic divisions to form the microspore tetrads. Clear to all of you? This much is clear? Okay. Now moving forward, as each cells of the sporogenous tissue is capable of giving rise to a microspore tetrad, each one is a potential pollen or a microspore mother cell. Okay, each is a potential pollen or a microspore mother cell. One second. Okay, done. Now each one is a potential pollen or a microspore mother cell. So the sporogenous tissue, you understood, no, this is capable of giving rise to microspore tetrad. So each one is a MMC or a PMC. Okay, the process of formation of microspore from PMC is called as your microsporogenesis, that goes through meiosis. Now the microspores, as they are formed, are arranged in a cluster of 4 cells, that is called as the microspore tetrad. Okay, microspore tetrad. As the anther matures and dehydrate, the microspores dissociate from each other and develop into pollen grains. Now inside, so I told you, no, they cluster because of callose, and their removal happened because of callase enzyme. Inside each microsporangium, several thousands of microspores or pollen grains are formed that are released with dehiscence of anther. Okay, released with dehiscence of anther. Done? This topic is clear. Next is megasporogenesis. We will come into that. I think we have to also go through a little bit of pollen grains and viability. Okay, viability. I hope that will be clear for all of you after that.